There you go.
There you go. Okay. So for anyone who's watching this now as being recorded, we've just welcomed everyone, and now we're just starting the recording. So for anyone who's just joining this by video, we welcome you too. So let me just, anyone else got any comments? Mitsu, Bernali, Akiko, anything else before we go? Okay. So again, I won't take too long on this piece, but just to tell you, so listen studies consists of people who are living with long COVID, people who are experiencing adverse effects after having had the vaccine, and other people, controls. We call them controls. We really mean are people who aren't suffering from either of those two things, because we really, in some of the studies, are going to need people who are in a similar situation to you with regard to their demographics, but are different from you in the sense that they're not suffering from a chronic condition. And so we're also encouraging many people within Listen to, if they can, identify others to join this who can serve as sort of that reference population. Someone said they're not hearing the audios. Are most people hearing the audio? I can see people, so you can just tell me. So sorry, Jose, but it seems like it's going. So right at this point, Kindred, by the way, has over 4,000 people who have joined Kindred. And I hope that's been a source of support for many people, as that information has been passed, and kudos to that team. And the Listen study, which you're part of, and that's what we're talking about today, has, believe it or not, over 2,100 people have joined the Listen study. And this is pretty novel in the sense that we didn't go to doctors and said, can you identify people in your practice who might want to join a study? And that there can be implicit biases in that, where some people are offered the opportunity to join a study and other people don't necessarily get the opportunity to join a study, and people may not come in through that pathway very well, or you're not at a center that's been asked to be part of the study, so you're really not possible for you to be part of that. But in this study, what we wanted to do was go straight to people and then let other people know, and through various mechanisms sort of just grow organically. And again, this study has to be worthy of your trust. So if people are having a bad experience, no one's going to join it. So people have to feel like this is something that's worthy of them being part of. Now, everybody came into Kindred and filled out a basic questionnaire to a variable degree of completeness. So one of the things I want to take the opportunity to do is to say we are doing things with the surveys, and I want to encourage everyone to be sure that they have filled out all their surveys. Plus, we're going to start getting to the point where we're going to send out more surveys specifically from the Listen study. So that's going to be an important part of what we do also. And we're going to be looking to you all as we begin to think about, like, what should we be asking? How do we make sure we capture it? How do we, in a way, advocate for what your voice is in this, what your experience has been, in addition to trying to advance the science to figure out how we can relieve some of the suffering, or most of the suffering that's going on out there. So this is another important piece. The connectivity to the medical records is another part. We're just going to start working on that as well. So many of you have connected your records. You know that Hugo is configured so your data doesn't move without your permission. There's no secondary marketplace. The data doesn't get sold. This has been, like, the whole idea was to give people agency over their own data, to be able to pull that data together, whether by wearables or health systems or insurers and payers. And so you're essentially have you accumulate those data assets and be able to share them into the study. This is a very unusual and novel effort here. And if we can prove that this can be done, I think it will spread as a mechanism to try to accelerate the way in which data can be aggregated, organized, and analyzed, so that we can learn in almost real time what's going on with people. And because the data streams, with your permission, we're able to use it within the study. And again, we did agree in the consent that at some point we might allow other investigators to use the data, but we would have to de-identify the data, but enable other investigators. But what won't ever happen is the data won't be sold. It won't be commercialized in that way. It'll be only used for science to try to advance knowledge, to try to make a difference to what you guys are facing, or what people that you care about are facing. And so that's the configuration of this. So we're urging people to make sure they fill out the whole questionnaire. We for some of the people, we're actually adding some additional questions that are being done, but we will be increasing, I think, the cadence of some of the information collection. And then the medical records are another important part of that. And then there's another piece of this study, which is that there's a smaller group that we are working on to gather blood and saliva so that it can be analyzed in Akiko's lab, where Nally's heading this effort to organize this. And it's collecting it in people's homes for a smaller group. And we are going to use that to try to correlate how people are reporting their experiences with what we can do from thousands of measures of the immune system and how it's functioning, to try to begin to make some progress towards understanding. And so quickly, I just also want to say that if I were in your position, I might be a little impatient. Like, okay, so when's stuff going to start moving out? The vibrations preprint is an example trying to move things out. We've got several other pieces that we want to move into preprint so you can see them. These are going to be basic descriptive studies that just help you understand who's in listen. And so we're going to do that for long COVID. We're going to do that for vaccine injury. We're going to do that for the combination. We're going to look at that all together and apart. And then we're going to look also people who have, like we've looked at vibration and not, we'd like to do that similarly for people. There are a lot of people who have tinnitus, ringing in the ears, and not, there's a lot of people who have POTS, so they have autonomic dysfunction. And we want to look at that group specifically. And we also are interested potentially in talking about people who have central nervous system issues, especially cognitive dysfunction, and how does that look? Now, there'll be overlaps in all these, but we're trying to characterize the group. So to manage your expectations, these aren't going to be Nobel Prize winning contributions. What we're trying to do first is explain who's in the study. What are some of these patterns? We're doing some what's called cluster analyses to see, I'll say like it's sort of what's the map? How are people organized with regard to their symptoms? Where are the areas where there's lots of people who have this cluster of symptoms versus this cluster? Because we believe that this label of whether it's long COVID vaccine injury is too broad. They're probably different mechanisms, but also different manifestations. Just because someone's called long COVID, you may be very unlike some other people who are also called long COVID. And the more specificity we get, the more likely we're to make progress. We start to build a taxonomy around this, a way to talk about this that's more sophisticated, more precise. And for each of you, it helps people understand what you're experiencing and not putting you under some big categorization that actually doesn't really capture who you are. So in sum, thank you. We can't thank you enough. Two, we're urging you to as much as you can participate with regard to filling out the surveys, to connecting your records, and if you're asked about those blood specimens, to give it some real consideration, that would be great. We're going to start pushing stuff out. Some of the stuff will be very basic in the beginning, and again, this idea of pre-printing. So it's an open access link. We'll send it out to people. And so there's no firewall, there's no price to it. It's just posted and it'll look like a paper, but it's there for public comment. It hasn't gone through peer review yet. It's just out there for your comments. So we're going to urge you to make comments and participate that way as well. And if anyone in the group wants to be more involved in studies or more involved in the things that we do, we need help. We're interested. We know you guys have got a lot on your plate just to deal with your health. So we don't want to impose or burden you, but I want to really extend the invitation that many people have different skills in this group that could actually help us advance and make more progress. So if you think there is a way that you want to be involved, then reach out to us and let us know. We for sure would want to hear that. And meanwhile, by the way, you also know there is a clinical trial we're doing with Paxlovid, 15 days of Paxlovid versus placebo. It has been limited to 3 states, but we're about to expand it out. Leslie sent out a note to Kindred members, I think today, to say we're extending it. I hope we're going to extend it to all. There are a lot of exclusion criteria. So it's like any clinical trial. It's like, there's certain ways you've got to satisfy to get in, but and it's only going to be 100 to start. But again, every one of these people is going to get deep immune phenotyping in Akiko's lab, both before, in the middle, and at the end of some of the data collection to help us understand whether, one, there are people who have maybe viral persistence. Are there people that are really responders to an antiviral like Paxlovid? And what can we learn in general about long COVID through this clinical trial? And I know there are others who may feel like either they don't qualify or they're not in the long COVID group, saying, when are you going to study us? And we're eager to. We just need to figure out, hey, brass tacks, we have to figure out funding. We have to figure out how to do it. There's lots of stuff to get together. But the more momentum we get, the more we can convince others that this is real. We need to invest in it. We need to get answers, and we need to help address the suffering. So with that, let me hand it over to Tiana, who will just talk a little bit about the preprint, and then she'll hand it over to Akiko.
I'm here to share a little bit about the preprint. I'll share my screen. So I want to make sure everyone can also see it. Can you see the PowerPoint? Okay, good. So this is one of the efforts that is ongoing in the LISTEN study. Like Harlan mentioned, there's many other avenues of questions that we're exploring, but this is just one of them. And this is specifically about internal tremors and vibrations as a symptom among people with long COVID and LISTEN. So the preprint is posted on medRxiv. If you want to take a deeper dive, feel free to look it up with this title on medRxiv and send us your comments or additional questions. As a caveat, some of the other results that I'm presenting today are more updated compared to what's posted on here. So if you hear something interesting today, but you don't see it on the medRxiv page, it's because we have some new analyses that we haven't posted yet. So the motivating question that we had was this question right here. Among LISTEN participants with long COVID, how do those with versus without internal tremors and vibrations differ from each other? And what's been described about internal tremors and vibrations has been this emerging definition of movement or sensation of movement at any location inside the body occurring with or without external visible movement or muscle spasms. And the scientific literature that I've been able to find were mostly focused around Parkinson's disease and essential tremor, but I also saw in the chat, I think Angie posted something interesting about this being a perimenopausal or menopausal symptom. So I would love to hear more about what are other situations where this has been described. All of this is contextualized with the caveat that clinicians, researchers, patients might have different definitions for what is a tremor and how to classify different types of tremor. And that's still being actively discussed in the scientific literature about the best ways to approach that. For our methods, we particularly focused on the survey data. And for the results I'm about to describe, we have not utilized any of the biospecimens data or the electronic health records data that you've shared. And the surveys, if some of you recall, asked about your demographics, socioeconomic characteristics, pre-pandemic medical and psychiatric conditions, as well as current conditions at the time of taking the survey. Current health status, which was measured by a standardized quality of life question, and current symptoms, choosing from a list and having the option to say other or none. And long COVID was defined by self-report. Do you think you have long COVID? Without imposing any additional criteria around when the long COVID symptoms started or how long those have lasted, the way that the WHO criteria imposes. And for internal tremors, the specific phrasing in the questionnaire was internal tremors or buzzing vibration. And if someone checked that box, then they were defined as having that symptom. So our statistical analyses took a two-pronged approach. We first just simply analyzed a bunch of variables between the two groups. So demographics, socioeconomics, all the conditions, and everything else that I have just mentioned, comparing them between the groups, the two groups, those with and without internal tremors. And two things I want to highlight here are that we also use the symptom questionnaire to calculate an approximation of something that the Recover Consortium proposed, which some of you might have read about in the news. So this is an NIH-funded research consortium that suggested a preliminary scoring system for how to define long COVID. And just out of curiosity, not because we explicitly endorse or have any sort of opinion about what they propose, we just wanted to check how their scoring criteria might or might not apply in the listen population. And we also defined something called new onset conditions. So that was calculated based on conditions that people reported as having at the time of taking the surveys, but were not reported as something they were having before the pandemic. The second branch of our analyses was a machine learning model. It was a gradient boosted tree machine. And the focus of this particular analysis was just to see what symptoms are important for differentiating people with internal tremors from those without internal tremors. Here are some of our results. In the overall group of listen participants with long COVID, median age was 46 years, a majority were female, a majority were white, and 37%, a substantial minority, reported symptoms of internal tremors or buzzing vibration as a long COVID symptom. And comparing the two groups, people with internal tremors were significantly more likely to be female, with no significant differences in age, race, marital status, pre-pandemic employment, pre-pandemic household income. Something really interesting that I want to highlight is that the two groups were not different in their rates of any of the pre-pandemic conditions that they had across 30-some pre-pandemic conditions that we asked about. But for infection characteristics, people with internal tremors were more likely to report an index infection during the pre-Delta wave. For health status, which was measured by the EuroQOL quality of life scale, we found that the overall score that people rated their health status on, for the overall group, the median was 49. And this is on a scale of 0 to 100. 0 means the worst and 100 means the best. And we found that people with internal tremors were statistically significant in rating their current health as being worse than the other group. And here's also the Recover score that I mentioned before. So it's an approximation because our questionnaire does not match the Recover Studies questionnaire directly in terms of the symptoms. But to the best of our ability, we approximated using their scoring criteria. And we found that overall, across everyone with long COVID in listen, we have a score of around 16, a median score of 16, interquartile range of 12 to 23. And people with internal tremors reported a median score that was higher than those without internal tremors. For new onset conditions, comparing all of these conditions, rates of mast cell disorders, neurologic conditions, anxiety disorders, and trauma and stressor-related disorders were significantly higher among people with internal tremors versus people without internal tremors. And again, this is new onset conditions that are occurring at the time of taking the surveys, as opposed to anything that was reported as a pre-pandemic condition. For the machine learning model that was created, here is a list of the top symptoms that were really important for differentiating the two groups. And I want to just highlight the top 5 right here. So hair loss was the most important, followed by floaters or visual lights, neuropathy, tinnitus, and sudden chest pain, and the other ones, so on and so forth on this list here. Some of the early conclusions that we can draw are that participants with internal tremors and vibrations, compared to others in listen who also have long COVID, they had similar demographics, similar health before the pandemic, but something about them might have been correlated with having different new onset conditions at the time of taking the surveys. and having worse self-reported health status measured by this quality of life scale. And the machine learning model helped us understand that it is possible to differentiate the two groups from each other on the basis of other long COVID symptoms that people reported. Like Harlan mentioned, there's a lot of other studies that are ongoing right now. And I want to specifically thank all of you on this call who are participating in Listen, and some specific people, including Sean, Kelly, and Teresa. I want to thank all of you. That's it. Feel free to write more questions in the chat and I'll try to get to them.
That was amazing. And for all of you on, Tiana's actually a medical student. And so we are so fortunate to have people at all sorts of different levels, but to get a medical student at this caliber to help us is remarkable. Akiko, do you want to take on next?
Sure. So I know that some of you have asked questions about what is the updated view of what might be causing long COVID. And so I wanted to just share a couple of slides as to how we are thinking about it. Let's see. Okay. Can you see this? Yeah. Okay, great. So yeah, as you have heard me speak about this before, we had 4 hypotheses for the root causes of long COVID. The first one being the viral reservoir or viral pathogen-associated molecular patterns, which is the nucleic acids of the viral genome. And these things can cause persistent symptoms because either the viral antigen that triggered T or B cells chronically, or viral RNA that can trigger this innate immune system to secrete certain types of inflammatory cytokines, can both lead to chronic conditions and symptoms. That was our first hypothesis. And this is really related to the SARS-CoV-2 virus that causes COVID. Second hypothesis was autoimmunity. This is basically T and or B cells that react against self-antigens. And we have seen a pretty striking evidence of autoantibody in severe cases of acute COVID. These patients either had prior existing antibodies against the immune factors that rendered them more susceptible to severe COVID, or they developed autoantibody during the course of infection that target various different tissues and cell types throughout the body. And once these things are triggered, it's very difficult to reverse them. And this is the second possibility as to why some people are suffering from the chronic phase of COVID. And the third hypothesis is dysbiosis of microbiome. Mainly, we were thinking about the gut microbiome and potentially causing some leakiness in the gut epithelium that leads to inflammatory conditions. And that's certainly been seen in ME/CFS and other conditions. We also thought of reactivation of latent viruses, that all of us carry a variety of viruses inside of our body, which are latent and don't cause any symptoms or disease. But when there is the immune pressure that's controlling these viruses, when they're lifted or when they're dysfunctional, these latent viruses can become reactivated. And the fourth hypothesis is tissue damage. This is doesn't have to even occur at the site of infection. We demonstrated using a mouse model of mild respiratory COVID that even a lung infection that's transient in the mice can lead to very long-term changes in the central nervous system that is not reversed even after weeks post-infection. And I sort of highlighted each of these boxes with different colors to indicate that throughout the research that we've been doing, we are seeing, as well as many others that are investigating the causes of long COVID, Viral persistence is one of the key hypotheses that still is supported by over 100 different publications. We still don't know if the virus that's persistent in some people are causing the disease, or it's just a sort of what happens with COVID and that it doesn't relate to the disease. And one of the best ways to figure this out is through this Paxlovid trial that Harlan already mentioned. What this allows us to do is kind of interrogate the immune system in people who have long COVID prior to the treatment with Paxlovid, and during the treatment and after the treatment, to see how the immune factors change over time. And how does that change correspond to benefit for the patients? Since we think that there are multiple root causes of long COVID, we're not expecting everyone to respond positively to Paxlovid, but we're hoping that a subset of people who respond to Paxlovid will tell us a lot about what are the biomarkers for responsiveness. What are the features that are common in the people who respond positively to Paxlovid? And in the future trials, can we enrich for those people with the biomarkers to provide a sort of better chances of people recovering or feeling better from long COVID with Paxlovid? So this is sort of an area that we're heavily investigating and are very excited to be able to do this with you all. Of course, we're recruiting some of you through the Listen study, and hopefully we'll be able to tell you what's going on once the study is completed. But that's a huge, I think it will gives us a huge clue as to what might be driving and whether the reservoir has any kind of real evidence for disease. Autoimmunity, I kind of graded out a little bit, but this I haven't really dismissed. We haven't dismissed this possibility. It's just that in our studies, we haven't found any common autoantibodies that are found in people with long COVID. If you look at the number of autoantibodies per person, they're not elevated necessarily in the long COVID participants compared to controls. And this is the study that Dr. David Petrino and our group have done together. It's called the My Long COVID study. And that has been on medRxiv for almost a year, but it's finally accepted for publication. So we should be able to share the latest with you shortly in the published version. But in any case, we don't see a clear-cut evidence for autoantibody against human proteins that are expressed on the surface or secreted from the cells. However, it doesn't mean that there isn't a T-cell mediated autoimmunity, and it also doesn't mean there isn't an antibody mediated immunity that targets intracellular antigens. So we're still very much open to this idea and investigating further. The dysbiosis, we haven't really been able to look into the microbiome yet, but we are looking at latent virus reactivation. And this is showing us that in people with long COVID, there appears to be an elevated level of Epstein-Barr virus reactivation, as well as potentially varicella zoster virus, which is the virus that causes shingles, that may be reactivating much more frequently than people without long COVID who recover from COVID. And this has been reported not just by us, but at least 2 other groups. And it's, again, we don't know whether this EBV reactivation is causing the symptom or it's just a sign of a dysfunctional immune response. But it is telling us there's some really biological difference in a subset of people with long COVID. And tissue damage, we're continuing to use this mouse model and developing other mouse models to be able to look at the causal role of these inflammation-induced damage. How do we reverse this? Such as the CNS impact and memory issues and brain fog and so on that's reported. So this is sort of like the updated version of what I told you before. And then, ideally, right, we should have clinical trial coupled with deep biological analyses for all of these going on. And there are some efforts going on, but I just wanted to kind of highlight, I'm sorry, the dog, what we're doing here. And then just sort of taking these insights into sort of the downstream effects and pathology that people are seeing. So the viral reservoir autoimmunity, latent virus reactivation, tissue damage, this may be kind of happening in concert within a person, or one may be driving the disease in a subset of people. We don't know what the actual sort of connection between these wheels are. But what we do know is that there is also evidence of downstream pathology, such as vascular damage, that's reported from Resipratorius and others, including microclots, as well as platelet activation. These are two events that we are investigating in Yale Lesson Study from participants who provided us blood and saliva. We're also looking at hormonal imbalance because one of the key defining factor that we saw in our long COVID participants was reduced levels of cortisol, as well as potentially other hormones. And so we're really looking into this hormonal imbalance as a possible downstream sort of mechanism. And mitochondrial dysfunction, this has also been reported both for ME/CFS and long COVID. We're investigating what the causes of mitochondrial dysfunction might be, and it may be triggered by the upstream events such as vascular damage and microclot formation, which would then impair the ability of cells to acquire oxygen and properly conduct the mitochondrial function in tissues. So these are sort of the downstream things that we also are looking at through the Yale Lesson Study. And of course, ultimately, we'd love to identify biomarkers for different subsets of diseases and, of course, therapy and cure ultimately, depending on what the driver of these diseases are. And so we are very excited to be able to do this with all of you. And, you know, we're working very hard. Bornali is working very hard in coordinating collection of biospecimen from a subset of you that's listening in today. So I'm going to just stop sharing. I think I took more than my share of time, but I'd be happy to address any questions, of course.
I think we always appreciate when you take more time. It's great. There are questions that are both in the Q&A, but some that were sent to us before. So this person, there are a couple questions we're going to ask for you, and then Bernali can answer one. But Akiko, this person said, I've been tested and treated for microclots. Is this something that's going to be more readily available in the United States? I've been paying out of pocket to see a doctor. What do you think about microclots?
Yeah, so we are also, as I just mentioned, very interested and are investigating microclots in people. And we are seeing definitely evidence of microclots and platelet activation in some people with long COVID, as well as some convalescent people who have recovered from COVID but have had COVID before. And right now, I don't want to make any claims that microclots are causing the disease because we don't know. We see some increases and it's variable. It's not that everyone has microclots. It seems to be a more activation in long COVID and even in post-vaccine adverse events. However, we need to be very careful. First of all, we need to have enough control to be able to say statistically significantly, is there an elevation? And in the people who have these microclots, what are the common symptoms and features, as well as immune markers do we see? So we are cautiously approaching this with proper controls and trying to understand what that means. We don't know what it means yet. There are places that are doing the microclot analyses. You know, I don't know which labs are doing them, but I know that there are some labs doing it. Hopefully they're doing it in a standard way, because there's also variation between each labs in the outcome of these assays. So yeah, it's very early days, and I don't know what to recommend, whether you should get the microclot assay and then what would be the treatment. And so we're still in the discovery phase.
Yeah, and I just endorse that also. Somebody asked me about this today in an email, and I said, I think it's promising. I know a lot of people believe in it. I'm not sure about both the validity, the reliability, that is test to test, place to place, because this isn't standardized. Like if you go get a potassium measured at one place and at another place, there are national federal standards that make sure that it should be the same. But we don't have that oversight right now on the microclots. And then ultimately, is it actionable? And some people think it is, and some people think it don't. I know how much everyone's suffering, so I don't want to say what people should do, because there's a bit of desperation in all of it. But just to say on the science side, it's not there that we could say that with confidence yet. A couple other people were asking Akiko whether the damage that they're experiencing now, do we think that it can be reversed? Is it permanent? And my view on it is I think you and I both heard stories of people who have been suffering for a long time, and then it does resolve and they've gotten better. We've heard other people who it persists in, but I don't think we can say yet for sure what would be considered permanent or what might be reversible. And I want people to have hope still, and there's definitely not evidence to suggest that what lots of people are suffering wouldn't go away. So I always try to encourage people to have hope. But I don't know, what do you think, Akiko?
Yeah, I think I really hope there is a way to reverse it. And certain tissues that are damaged, of course, depends on which organ and how replaceable those cells are and so on. But I mean, that's why we're doing this research. We really want to find something that would be helpful. And we are doing, because we have these mouse models of tissue damage, we are able to try all kinds of different agents to be able to reverse the damage. And that's what some members of my lab are doing right now. And we'll let you know, if we find something, but we're doing this in a sort of way, in which science has approached similar kinds of inflammatory conditions before with the drugs that are able to kind of restore and replenish the damaged cell types. So I'm hopeful. Of course, we're not going to promise anything because, that would be irresponsible. But yeah, I really hope that we can find something.
We know that some days it may be hard to have hope when you're facing all these things. So we want to encourage people not to give up and to feel maybe we can together all find answers. Bernali, you answered this on the chat. Some people put it in the Q&A. I know, maybe you could just say out loud, people listen who have had the deep immunophenotyping, maybe you can make a comment or two. I'll just say to folks that it's not usual for us to be allowed to return research level results to participants. We want to be able to do that, but the regulatory bodies are concerned about it. By the way, microclots is an experimental test as well, but because clinicians are ordering it, they can give it back to you. But when we're doing experimental tests in the lab that for research, there's a reluctance because of a fear that people will act on them in ways that aren't yet, proven out that even the tests should be acted on. But we think we found a path to share something. So Bernali, do you want to talk about that?
Sure. So we right now have approval from the IRB to share research-only reports, and we are in the process of getting the reports ready, finishing off all the assays first, and then getting the reports ready. And once we have them ready, we will reach out to you because that's the protocol that the IRB has proposed for us. We'll send out a mass email to anybody in the study who has contributed biospecimens to our study. And once they acknowledge and request for the reports, we'll individually be sending out those reports. But those will be research-only reports, not actionable. Thank you.
A couple people, Akiko, were asking about Epstein-Barr virus and the reactivation, and then somebody asked whether or not long COVID. They asked whether it can reactivate other viruses as well, but I think we're thinking like maybe those are the actual cause of the symptoms. But do you want to just say a minute, what are you thinking about Epstein-Barr virus now? As I know you mentioned as a mechanism, this reactivation of viruses, but in your lab, you're following this stream, right? As a way to sort of figure this out.
Yeah, definitely. So we are testing experimentally whether SARS-CoV-2 infection can reactivate EBV in latently infected B cells, for example, in the tissue culture. So we can do this just an in vitro experiment to see whether there's any interaction between the viruses, and that would be a direct path to reactivation. But an indirect path would be that the SARS-CoV-2 infection led to impairment in T cells or B cell immune control of these latent viruses, and that would be sufficient to reactivate. So that's sort of the cause for reactivation, but also the consequence of reactivation, whether EBV treatment against EBV, reacted EBV or monoclonal antibodies, or there's some other form of treatment to deal with the reactivated virus. Would that be helpful to people? And these things would obviously require some, form of clinical trials to be able to understand the benefit of these drugs. But yeah, so we are still investigating, what causes it and what is the consequence of it.
Somebody was asking, how can you tell that it's reactivated?
There are two separate evidence. So one is the Cell paper that was published by Jim Heath's group last year, where he was able to detect viremia, which means the EBV viral DNA was found in the blood of people during the acute phase of COVID. And when they follow these people over time, the ones who had viremic EBV were the ones that were more likely to develop long COVID versus those who didn't. So this was a longitudinal study that Jim Heath's group has done. And there are Mike Peluso, as well as our lab, we found elevated antibodies against lytic protein of EBV, which is normally hidden in latent cells. They don't express these proteins during latency. The fact that you have elevated IgG against these things are quite indicative of recent reactivation with the virus. So we're not saying that people who have these antibodies have live infectious EBV floating around in their blood. We actually can't find, viremia in these people. However, we do see evidence for recent reactivation. So this may have happened during the acute phase of COVID, or it may have happened a few months ago. We don't know. We don't have the longitudinal samples to tell what happened in the intervening time period. But yeah, so that's the evidence we have.
Well, that's great. Somebody's asking about autoantibody testing that doctors can do. And it may be just an opportunity to reflect on this paper that's going to come out in Nature, where there was a panel, more than 2,000 autoantibodies that were tested. And I don't know if you want to just say something about that, Akiko. I mean, sort of that we weren't able to identify much there.
Right, exactly. So this is the REAP strategy that was developed by Professor Ring's lab. And it contained actually over 6,000 antigens from human.
6,000, oh, wow.
Yeah. And of course, there are, several autoantibodies that are found in people with long COVID and people without long COVID and the healthy control. So, and in fact, all of us carry some form of autoantibodies against, several autoantigens, but they're not pathologic antibodies. And so, but even though we didn't find it in that panel, like I said, we haven't excluded the possibility that autoantibody against internal antigens, intercellular antigens, or some nucleic acids or something else may be causing the symptoms. So even though, that REAP didn't find a clear pattern of autoantibody, we certainly haven't given up on that hypothesis.
And somebody was raising an issue to us, asking about that study was about long COVID. We also are going to be doing the same kind of analysis for those who are experiencing vaccine injury. I will say one thing about this issue of vaccine injury, because somebody, I think, who had come into the study with long COVID was wondering, well, why are you studying people with vaccine injury? And I want to say that it's clear to us that there are lots of people who are suffering, and there was a lot of overlap in terms of a chronic condition that had really unraveled people's lives. And it's important to know that these people who are reporting this about the vaccine, that most of every one of them that I've talked to, like, doesn't want to get involved in the politics of this. They're not anti-vaxxers. They got vaccinated. They are people who have found themselves in a position where it's inconvenient for anyone to even talk about them because of the way the politics occurred. And if people with long COVID have found themselves in positions where people don't believe them, I can say it's even more intense for these individuals who are reporting symptoms after vaccination. And so, I think when Akiko and I, became aware of this and started talking about this, we recognized that there was some risk because of the political maelstrom about this. But we wanted to approach this scientifically, and we wanted to approach this humanistically. I mean, people are suffering, and we've developed a platform. We've developed a way to work together with folks, and we thought that we should be, we should be willing and interested, and we are, in trying to find answers there too. So, what really I think in the end was there will be different mechanisms, no doubt. When we're talking about viral persistence, that's not going to be the thing that we'll find that occurs for people who had problems after vaccination. And I want to say, I believe, and I know Kiko believes, vaccinations are a modern medical miracle. Millions of lives were saved because of the speed with which vaccines were put out. But it can be true that there still were some people who were harmed by the vaccine. Both of those things can be true. The net value of it was immeasurable. And yet, there's still some people harmed. If you look at vaccines over the history, it's almost always true that there's some things that happen when people are vaccinated. In this case, rare relative to benefit, but important to anyone who's affected. And I think our job is to understand, how can we prevent in the future? What is this? And how do we help alleviate suffering? And again, we're trying to take this in a broad-based way. So, long COVID, vaccine injury, it may unlock some of the mechanisms by which the immune system ends up conspiring against the best interest of the host, of the person. And so, we're hoping that this all helps unlock this. Interestingly, when we measure the health status in Listen of people who are reporting vaccine injury and of people reporting long COVID, and by the way, even the long COVID group, we're not requiring people have had a positive test. We know lots of people got this before tests were available. We're accepting what people say. And when you look at their health, both groups are suffering about equally. That score of 0 to 100 shows severe disabilities, severe illness in both groups about equally. Maybe we can use them to help us understand each other, because they can almost serve as controls for each other. Because like I said, there's some things that would not have happened otherwise. But anyway, I just wanted to mention, so we're just about at time. Let me just thank everyone. We'll look through and see if there are other questions that we can answer that we missed. I'm going to give Akiko the last word here, but I just want to say, from the depth of my heart, I want to thank all of you for being part of the study, being part of the team, being willing to come along. We are always interested in your comments, and not just positive comments. Tell us what we're doing wrong. Tell us what we can do better. Tell us how we can, somehow be more responsive to this, how we can accelerate this. And also great deep thanks to the Listen research team at Yale, who've spent countless hours trying to move this stuff forward. And Akiko, let me hand it back to you for the last comments.
Yeah, thank you, Harlan. Yeah, I'm really grateful to be part of this large team, including so many participants here today, to be able to investigate this. I think this is already revolutionizing the way we do science, and much thanks to Harlan and his vision in allowing this to happen. And we're very much committed to figuring out what's going on with people with long COVID and people with post-vaccine adverse events. And we have collected the first set of samples from people with vaccine injury, and we are just waiting for the results to come back. And so hopefully next time we can share some of the collected data from that study. But we're very, very much dedicated to doing this. And we apologize for the slowness of the studies. Even though we're doing everything at lightning speed, things take a long time, even with just the regulatory paperwork alone. So it's really, it's not an immediate answer that we can give you, but just know that we are really working hard, and we thank you all.
Thank you.